Good morning. Welcome to Biology 1302. Moving on, we're going to discuss Chapter 9 today, Sexual Reproduction and Meiosis. This will cover DNA, inheritance, and biotechnology in Unit 2, Sexual Reproduction. Asexual reproduction is relatively rare. It occurs in some organisms. You have an original cell that will reproduce to produce a daughter cell, and you see it's pictures of a four-step process. You see the duplicating cells starting to divide, and then you have two cells that are the equivalent of the original. Again, this does happen. It's quite common in some organisms like bacteria. They undergo a process that's sort of an asexual reproduction. It's called binary fission. There are also some fungus organisms that reproduce this way. So again, asexual reproduction is a type of reproduction that does not mix genetics of two different individuals. Mitosis underlies asexual reproduction. We talked about mitosis. The problem, the issue with asexual reproduction is you are producing a genetically identical offspring. This just happens to be an amoeba, and again, you have two identical daughter cells. Give them a few more minutes, and they will begin to take on that same shape. Meiosis is required for sexual reproduction to occur. Meiosis is a type of cell division that generates eggs and sperm, each of which is genetically unique, bringing about all new genetic combinations in offspring. These specialized sex cells lie at the heart of sexual reproduction. Here we see a nice little, looks like an Australian shepherd and all of her cute little puppies. Attracting a mate, producing sex cells requires a lot of energy, which is costly to the organism, so genetic diversity is worth it. It is worth spending all of that energy to ensure genetic diversity in your population. Sexual reproduction is worth the cost because it creates and maintains genetic diversity. Later on in the year, we will talk a little bit about the problems that can happen when certain organisms or certain populations become don't show enough genetic diversity, and genetics can kind of bottleneck, and that can cause some problems. Homologous chromosomes are matched sets. You see, you got a pair that are the same size, they're the same length, have the same kind of genes in the same places, but this one is red, this one is blue. So you have some different information. You know, this is hair, for instance. This would be hair color, eye color, ear shape. Hair color, eye color, ear shape. But again, same length. So that's a homologous pair, simply different information. Sexually producing organism consists mainly of diploid cells. These are cells that contain 2 copies of chromosome 1, 2 copies of chromosome 2, 2 of 3, et cetera, all the way down to where we have an X, 2 X's for this individual, or an X and a Y. The 2X individual would be a female, the X and Y individual would be a male. This image at the right shows what is called a karyotype, shows all 46 chromosomes divided into 23 pairs. 22 of these are called autosomes. The 23rd pair are called sex chromosomes, and again, you have an X for females and an X and a Y for males. Homologous chromosomes are not identical. They carry the same information, but the specifics that are going on in A and the specifics that are going on in A over here may vary. They may not be the same thing. So you may have different, while it's still controlling the same thing, the specifics of those are going to be different. Each person inherits one allele, so you get one of this from your mom, maybe this one comes from dad, and then these are duplicated, mom and dad. This is replicated chromosomes. Oh, actually it even says from the mother and from the father. Life cycles, again, we talked about mitosis earlier. This is meiosis. This is a mitosis reproduction here, of where you're just getting the same cell over and over and over. Then you wind up getting what's called development. Then you get to meiosis. And again, we're going to if you'll notice here, there's 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7, 8. But there's a blue one and a long blue, long red, short blue, short red. Then you get over here, when we've made the eggs or sperm, we cut that down in half to where there's only one of the long ones and one of the blue ones in each cell. Human males and females undergo a process of meiosis. The original cells are referred to as germline cells, germ cells. They undergo meiosis to produce eggs and sperm. Once we get to the gametes are not going to divide any further. You have a sperm cell, will encounter an egg cell. Here, fertilization will occur. Again, gametes are haploid. If we go back to here, originally, before duplication occurred, you had the one long blue chromosome and the one long red one, and the one short blue one and one short red one. And then as you see in these four down here, cells down here, only one of each one of the longs and shorts are found in the gametes. Fertilization is going to occur. The first cell of a new organism is referred to as a zygote. Here's the fertilization step, and the zygote is now this was a one-end cell. This cell only has 23 chromosomes. This cell only has 23 chromosomes. Once they have fertilized and it's a zygote, we now have the 46 chromosomes. Then we're going to undergo mitotic division to fully develop the little baby. We're going to continue the mitotic divisions until we become fully adult humans. The fully adult humans will continue to undergo mitosis because they have to make new skin cells, they have to make new hair, they have to make all sorts of wonderful things. But some cells are now undergoing meiosis to produce sex cells. The zygote is a diploid cell. Again, each one of these, only 23. So that is a haploid cell. The zygote has the full complement of 46. So it's going to be a diploid cell. The zygote then, again, undergoes mitosis to develop into the baby. Haploid gametes give the offspring the right number of chromosomes. As I said, each one of them is going to have 23. So the zygote, once it's here, is going to have 46, two sets, A and B, as they get ready to start mitotic division. Meiosis is very similar to mitosis. You're going to see some names that are very familiar. In meiosis 1, as just a quick overview, you're going to see you have the pair of large blue chromosomes, the pair of large red ones, the pair of small blue ones, the pair of small red ones. And then when we divide, we only see one pair go through meiosis 1. And then we're going to go through meiosis 2. And now we've had this to, we started out with a cell that had 4 chromosomes. We now have a cell that has 2. I think I covered all that. Meiosis involves 2 divisions. Again, meiosis 1, you're going to separate you're going to have the number of chromosomes. Meiosis 2, you're going to separate into 4 individual haploid cells. Germ cells divide in meiosis 1. So the DNA replicates very similarly to stage 1 of mitosis. This will be a prophase-like stage. Then you're going to see a metaphase-like stage where everything's lined up in the middle, but instead of being lined up all in one line, you see they've lined up in pairs. Then these are going to separate. There's the cleavage furrow, the contractile ring, and you move over, and now you have a more recognizable metaphase and go through all the other stages, get to the end. And we're going to go through each one of those as we move along. So in prophase 1 of meiosis 1, again, very similar to prophase of mitosis. See the nuclear membrane starts to break down, the centrosomes start to form, the centrosomes attach and connect the sister chromatids, the centrioles start to form, the spindle fibers start to form, and there we're into the late part of prophase. In metaphase 1, again, prophase 1, homologous chromosomes pair up. Yeah, they come together. It's either twisting and combining and condensing. Here, they have paired up into their homologous pairs. Then in metaphase 1, again, and a human example of this would be like maybe the red one is the chromosome number 2 you got from your mom, and the blue one is chromosome 2 you got from your dad, and then this red one is chromosome 5 you got from your mom, and this is chromosome 5. So all the 1s are going to line up, all the 2s are going to line up, 3s, 4s, 5s, etc. That's what's going to line up. They're going to line up in what are referred to as homologous pairs. All the 1s, all the 2s, are the 3s. Anaphase 1 of meiosis 1, you see the pairs have now, instead of individual chromosomes moving toward opposite sides, we have the whole pair moving. Again, the contractile ring, cleavage furrow is going to form, and we're ultimately going to wind up with 2 cells. Again, telophase 1 and cytokinesis is going to happen very quickly, and you can see it down here again at these cells. Then we have 2 cells. In meiosis 2, we now have formed 2 cells. Each one, if you'll notice, has a prophase stage. This drawing is a little bit deceptive in the fact that as a cell moves into prophase 2, it doesn't reform a nucleus. These things don't completely break down. This is just an artist's way of showing you that the process is going to start over. The metaphase 2, okay. Metaphase 2, you see now a more traditional look at what more like what metaphase looked like in the mitosis stage. Once again, in anaphase 2, the different chromosomes are now going to separate, with 2 of them going this way, 2 of them going this way. Same thing happening in this cell. Again, contractile ring, cleavage furrow forms, and ultimately you wind up with 1 diploid, with 4 haploid cells, all produced from 1 diploid cell. The big thing to remember about meiosis is this is going to be a halving of the genetics. You have cut the chromosome number in half. This cell had 46 chromosomes, each one in human beings. Each one of these only has 23. Meiosis is a process that generates variation. The reason not all siblings look alike is because of meiosis. Again, here's the nice little puppies, and according to our knowledge here that's given us, these pups all came from the same parents. They're in the same litter. But if you'll notice, you know, this one and this one are sort of pale and speckledy. This one's a little darker, but it's still got the speckled. This one's almost completely black with a little white collar. This one's more of a brown color with a little white collar. So all of those different relations, all of those different traits are the result of the different shuffling that occurred. Mom produced a bunch of eggs, Dad produced a bunch of sperm, and this sperm and this egg got together to produce one group of combinations, this one another one, this one another one, this one another one, and that one yet another. Something that can happen during prophase 1 to even further increase the genetic diversity in populations is something called crossing over. In crossing over, during meiosis 1, homologous chromosomes pair up precisely, 1 from mom, from dad, with 1 from dad, 1 with mom, 1 from dad. But during crossing over, pieces of those homologous chromosomes can change place. If you look right here, what happens is this one is still the original form, this one is still the original form, but these two, a little section got tangled up, and the little AB section of the red one is now here, and the AB section of the blue one is now there. So, same information, just different specifics. Same genes, just different genetics. And again, this is called recombination. because we're getting new combinations of alleles. Now, instead of having two chromosomes, two identical chromosomes here and two identical chromosomes here, we now have 4 unique chromosomes formed from that one original cell. Another thing that can happen in metaphase 1, this one can't, the difference is crossing over may occur. Independent assortment absolutely occurs. And what that means is that independent assortment you see here, as you got, they've now labeled your chromosomes 1, 2, and 3. You're going to make all these different, combinations. Here, this one, you have 1, 2, and 3, 1, 2, and 3. But here you have a different combination. Over here, you have even another combination. Basically, what this is telling you is these things are going to line up at the metaphase plate, but just because your dad's chromosome is over here doesn't mean all of your dad's chromosomes are over there. And just because moms are here doesn't mean all of moms are going to be here. You could have mom, mom, dad, 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 mom, mom, dad, dad, and up here you could have, other combinations. So as those things start to form the gametes, you're going to wind up with even further amount of genetic diversity, not only within a population, but within a family. How many of you know somebody where, parents have 5 or 6 kids, and the kids all none of them really look much alike. There's some tall ones and some short ones and some blondes and some brunettes, green eyes and brown eyes. That's because of independent assortment. In one mating, any of a mother's possible egg cells can combine with any of her mate's possible sperm cells. So every time eggs and sperm are made, that combination is going to be different. And human beings and most mammals practice what is called random fertilization. The sperm that contains the blue-eyed allele is just as likely to fertilize the egg as the sperm that contains the brown-eyed allele. Monozygotic twins. One thing that can happen in sexual reproduction is the production of something called twins. Again, we all know that dogs have litters and cats have litters and rabbits have litters, but human beings are actually designed to have one child at a time. If you look at the structure of animals like dogs and cats that have litters, their uterus is shaped very differently from the human female uterus. But if you look at other animals that are designed by nature to have one, such as cows, horses, or elephants, those uteruses, while much bigger than the human uterus, the general shape is the same because that uterus is designed to hold one offspring. However, sometimes we have some things where monozygotic twins are genetically identical to each other. These are what we refer to collectively as identical twins. In early development, here's the egg, here's the sperm, fertilizing a single egg, producing a single zygote. But at some point in gestation, a separation occurs to where you have 2 individual organisms developing from that one fertilized egg. These 2 young men right here are going to share the same genetics. This guy grows up to be a bank robber. This guy grows up to be an attorney. This guy robs a bank. He can blame it on his brother if the genetics is there because they are genetically identical. There actually was a case, where was it? Texas, I believe, a few years ago, where a pair of identical twins, I think this was on Maury Povich or Geraldo Rivera or something, but these were a pair of identical twins. One of them is serving time for a crime that he insists that his brother committed, and the fact that DNA was found, they could not exclude either one of them. And since one of them said he did it first, I guess, he's the one who got prosecuted, and he's sitting in jail or in prison 25 or 30 years for whatever the crime was, and again insists that it was his brother who actually performed or did the deed. Dizygotic twins have their own unique DNA. Dizygotic twins are the result of two different eggs being fertilized. Now, I had a student many, many years ago ask me this, and so I'll just throw it out there. Monozygotic twins are always going to either be two boys or two girls. Monozygotic twins are never going to be one boy, one girl, because they have to develop from the same egg. Dizygotic twins, however, are oftentimes one male, one female, or one boy, one girl. Meiosis and mitosis have different functions. Again, mitosis adds and replaces identical cells. So when you're making bone tissue or muscle tissue, that is a mitotic process. That's mitosis. When you're making eggs and sperm, that is mitosis. Mitosis produces haploid gametes. They do have some similar processes, however. The process of crossing over and independent assortment makes meiosis work differently. Again, these processes the process here, crossing over here, independent assortment, don't take place up here. So they make them work differently. Errors can occur in meiosis. If chromosome fails to separate properly, gametes may end up with the wrong number of chromosomes, leading to abnormalities in offspring. Failure of chromosomes to separate properly is called non-disjunction. And a classic example of non-disjunction is something called trisomy 21. 3 copies of chromosome 21. You'll see there's only 2 copies. That's the normal karyotype. The karyotype showing 3 chromosome 21s has been inset right here. This is commonly referred to as Down syndrome, and there's a young man right there who suffers from Down syndrome. Again, trisomy 21, Down syndrome individuals have 1 chromosome too many, 3 copies of chromosome 21. This is one of the very few chromosome abnormalities to do with autosomes. And remember, all of these, except the X and the Y, are autosomes. And Down syndrome is one of the few autosomal disruptions in number where the offspring will actually live into adulthood. Generally, if something happens and these things have been added or deleted, that offspring is not going to live. If it's born, if it comes to term and is born, it's probably not going to live very long. Non-disjunction occurs in anaphase 1. It causes abnormalities in all gametes. Again, here you see what should be happening and what you wind up with. The gametes here formed this way. The little blue line is all that this sperm has. It should be encountering a red chromosome in each one of these gametes. So you wind up with a red. What you should get is a red and a blue, a red and a blue, a red and a blue, a red and a blue. But what you're getting is this pair of zygotes that have too much DNA and this pair of zygotes that don't have enough, because all this one has is what was in the sperm. Non-disjunction, if it occurs in anaphase 2, can also cause abnormalities in some of the gametes. Again, same situation that you're showing here of these not separating ultimately, but you wind up again 3 there and only 1 there. Non-disjunction can be lethal. Again, it is normally lethal. Again, you've got the same issue. Chromosomes tend to make development impossible when you have too much or too little. There are some non-disjunctions of sex chromosomes that can cause some sexual disorders. These individuals tend to have low IQ, as much as failure to develop secondary sex characteristics. There's actually one of these that this does not apply to, and that is this one. Triclo X occurs in 1 in 1,500 females. The non-disjunction factor here is that one of the pair of X chromosomes in egg or sperm production didn't separate, so the offspring is born with 3 X chromosomes. Again, these people are structurally female. The XXY or Klinefelter syndrome occurs actually more often than you would think, 1 in 750 males. Again, the 2 X's didn't separate. Here you have 2 Y's, Jacob syndrome. I'll say a little more about Jacob syndrome in a minute. But again, 1 in 1,000 males. And then Turner syndrome, where what happened here is the 2 Y's in sperm formation didn't separate, but the sperm that actually fertilized the egg was the sperm that didn't get a Y at all. So, again, XXY, XYY are males, XXX, XO are females. Think about Jacob's syndrome. When I was taking these classes years ago over at Euler, and we're talking 25, 30 years ago, this was actually called the Superman genotype. And it was believed at the time that that extra Y chromosome gave men who had that a definite advantage over other men. It gave them more testosterone. They were, inherently stronger. They had more drive and more ability. They basically said that people like Bill Gates and Steve Jobs and Mark Zuckerberg and the guy who founded whatever Twitter and the Google guy and the Facebook guy, all of them would probably be examples of people who had the XYY because they were able to do those things because they had the extra Y chromosome. That was one side. The other side of it was they might also grow up to be mass murderers like Ted Bundy and some of the other mass murderers that we've seen. Ultimately, in the last 25 years, all of those things have been debunked. These people right here, the XYY men, are no different from a standard XY man. They don't get any advantage from having that extra Y chromosome. And again, some research is now saying that they may actually tend to cause some problems as well. Wraps up chapter 9. That actually went a little faster than I thought it would. If you have any questions, please email me, trussell@uaptc.edu, or in the contact section, course messenger in Blackboard, send me a message there. If you have any questions, you should have a new exam opening up in the next few days. Make sure you pay attention for that. And if you need anything, just send me a message. It's Friday where I am, so if you want, everybody have a great weekend. If you're watching this late on Sunday night, get a lot of rest, and let's hit a new week starting on Monday.